So in dark reaction, what happen? Every time there is a substrate. The name of the substrate is RUBP, ribulose biphosphate. So let me tell you more clearly what RUBP do, or what exactly the function of RUBP. Actually, RUBP is a type of compound. So as we know that RUBP is acceptor of carbon dioxide. So what happened? One molecule of carbon dioxide. So every molecule of carbon dioxide have one atom of carbon. It combines with RUBP, ribulose biphosphate. It's a compound. So the number of carbon atom is one here, one carbon atom, and number of carbon atom in RUBP is 5 carbon atom, right? So there are total 6 carbon atom. When they both will combine, they will form 6 carbon atom. So they form two molecule of, look, you can see over here, PGA, phosphoglycerate. They form two molecule of PGA, that is phosphoglycerate. What happened, Affaf? Phosphoglycerate is 3 carbon compound, but here formation of two molecule takes place. So number of carbon atom are equal at both of them, right? So this RUBP is the primary acceptor of carbon dioxide. Did you got my point? Is that clear? And if someone asks you that which is the first stable compound of C3 cycle, and this is the 3 carbon compound, right? That's why you call it C3 cycle, and it was discovered by Kelvin, so you call it Kelvin cycle, right? This is 3C cycle.
Sir, sorry, when you came to the 3C part, you weren't audible to me.
Okay. So look, here what I am saying, that one carbon atom is added by RUBP. RUBP itself has 5 carbon. So total number of carbon atom, 6, right? RUBP combined with carbon dioxide. And what is the name of that enzyme which help the combination, or which help RUBP to combine with carbon dioxide? The name of that enzyme is Rubisco. Last class we have discussed in detail that Rubisco is ribulose biphosphate carboxylase oxygenase, right? And this is the most abundant enzyme or most abundant protein on the earth, right? So in the presence of Rubisco, RUBP and carbon dioxide, they combine to total 6 carbon atom. Due to that, the formation of two molecule of PGA takes place, and PGA is 3 carbon compound, right? So there are total 6 carbon compound. So first stable product, because PGA phosphoglycerate is a stable product. So first stable product of this cycle is 3 carbon compound. That's why we call it C3 cycle. And it was discovered by Kelvin Melvin. That's why we call it Kelvin cycle, right? Is that clear?
So why is it called C3 cycle?
Because you know the first stable compound is 3C, PGA is 3C compound, 3 carbon compound. That's what we call C3 cycle.
Okay.
Right?
Okay.
Okay. If you want, you can note it down. Please note it down. In Kelvin cycle, note down this statement. In Kelvin cycle, the first stable compound in Kelvin cycle, the first stable compound is first stable compound is sorry, in Kelvin cycle, the first stable compound is PGA, phosphoglycerate, which is a 3 carbon compound, which is a 3 carbon compound. That's why which is a 3-carbon compound. That's why it is called, that's why it is called C3 cycle. That's why it is called C3 cycle. Next, now look, you can see over here that this carbon dioxide combined with RUBP, ribulose biphosphate. Then two molecule of phosphoglycerate are formed. And now what happens? 2 ATP breaks down into ADP, means generation of energy takes place and phosphate gets released, right? So here ATP become ADP and energy is released. Again, NADPH breaks down into NADP. So this complete process is oxidation process, right? Energy consumption takes place. Then after that, due to this process, due to expenditure of ATP and NADPH, formation of 2-glyceraldehyde 3-phosphate takes place. That is called in short G3P, right? Formation of G3 takes place. G3P is again a 3-carbon compound. 2 molecules of 3-carbon compound takes place. So total balance of is still 6 carbon, right? So out of these 6 carbon, 1 carbon is released for glucose formation. Now out of these 6, when 5 carbon remains, the generation of a product takes place or a outcome comes, that is called ribulose phosphate. So formation of ribulose phosphate, that is a 5-carbon compound. Out of these 6 carbon, 1 carbon has been donated for the formation of glucose, right? Then from ribulose phosphate, again 1 ATP expenditure takes place. ATP get breakdown into ADP. Again, energy is released and phosphate is released. Now one more phosphate molecule is added over here, that is represented by yellow color. Now the again formation of ribulose biphosphate takes place. Hence the generation of ribulose biphosphate takes place. So what exactly this 5-carbon compound have done? It have picked up 1 carbon atom from the carbon dioxide and donated this carbon to glucose formation. As we know that glucose have the 6 carbon atom. So this Calvin cycle have to go for 6 times for formation of 1 glucose molecule. This is all about the Calvin cycle. Is that clear?
Sir, can you explain the third part? I'm having trouble understanding it.
Third part, I means regeneration. So look, here there are 2 molecule of G3P, right? So G3P, that is a glyceraldehyde 3-phosphate, right? 2 molecule of G3P are formed. When these 2 molecule of G3P, so there are total 6 carbon. Out of these 6 carbon, 1 carbon has been donated for the formation of glucose. Now from 6 carbon, 1 carbon gone. So the remaining carbons are 5 carbon. So due to that, the formation of a 5-carbon compound takes place. We call that ribulose phosphate, RP, right? Not biphosphate, ribulose phosphate. It have, look, you can see that it have a purple color 5-carbon atom and 1 phosphate. We call it ribulose phosphate. Again, here, again, ATP breaks down into ADP, means energy is released, and along with the energy, a phosphate is released. This phosphate combined with the ribulose phosphate, and due to this, formation of ribulose biphosphate takes place. This is called the regeneration process. So what I say that what RUBP do, RUBP is a 5-carbon compound, right? It takes 1 carbon from the carbon dioxide and it deliver to the formation of deliver the carbon for the formation of glucose. That's why at every cycle 1 carbon is delivered by the RUBP, right? And for the formation of glucose molecule, total 6 carbon are required. So the Calvin cycle have to go 6 times for the formation of 1 mole molecule of glucose. Is that clear? Affaf, you are not audible. Is that clear?
So
I'm unable to listen you.
there are six carbons
I think there is a poor connectivity, Affaf, because your voice is breaking.
so glyceraldehyde, whatever that is.
Uh,
One is given to glucose and other carbohydrates, right?
Let me message you. Actually,
Oh, okay. Can you hear me now?
hello, Affaf.
Okay.
Yeah, you're audible to me.
Hello?
Yeah, am I audible to
Yeah,
you?
I was,
Yeah.
yeah, you are now. I just changed my internet.
Okay. Okay.
So,
Now you're audible to me.
okay.
Yeah.
So I was asking the third part, you said there are six carbon um, molecules, right? One is given to glucose and other carbohydrates.
What is
Is that
given
right?
to? What is? Right. The six carbon atom. There are two molecules.
One goes to glucose and other carbs, right?
Right. One goes to glucose, and another five, they again regenerate. Means again the formation of ribulose phosphate takes place.
Yeah.
And ribulose phosphate changes into ribulose biphosphate. Right. So that's why we call it regeneration process. So every time this ribulose, what ribulose biphosphate does, it take a carbon atom from carbon dioxide and deliver to glucose, right? And for the
So now
formation
you're
of,
not audible to me.
I'm not audible to you.
Yeah. Yeah.
Actually, I'm using high speed internet connectivity. Optical fiber I'm using. So might be there some internet connectivity at your end.
Yeah, it could be. But now I can hear you.
Look, what I'm going to do, whenever I'll speak, I'll switch off the camera, because by this way, the data consumption will reduce at your end. Like downloading data downloading will decrease at your end, so that will provide you more connectivity. I think now it should be more clear. Is that?
Yeah.
Yeah, actually, video consume more KB. Fine. So now tell me, is that crystal clear to you? Did you got all the points?
Uh, I can't say anything about the third part.
Third part, is that not clear to you?
No, not yet.
Okay, no issue. I'll tell you again. Look, what happens here? I think so. Is it clear till here, till this point? Glyceraldehyde G3P, and how does
Yeah.
the one
Yeah,
molecule is
one of the carbon goes to glucose and other
right.
carbohydrates, right?
So out of these six, one goes. How many carbon remained? Six minus one.
Carbons.
6 minus 1.
One goes to glucose, so there's five.
Right, five. So with the help of those 5 carbon and 1 phosphate, formation of a molecule takes place from G3P. Formation of molecule takes place, and the name of that molecule is ribulose phosphate. Got my point?
So 5 carbons and 1 phosphate makes ribulose phosphate.
Exactly.
Okay.
Right. Again, ribulose phosphate is not carbon dioxide acceptor. Right. So what happened? One ATP breaks down into ADP.
Okay.
Right. We know whenever an ATP breaks down into ADP, for formation of ADP plus 1 phosphate, 1 inorganic phosphate released, right? This one,
Yeah.
right? So this phosphate is added in RP, ribulose phosphate. 5 carbon plus 1 phosphate was already there. One more phosphate added. From where it came? It came from ATP. Got it?
Yes. Yes.
Okay. So what is the name of ribulose biphosphate? Bi means 2 phosphate. So we name it ribulose biphosphate. So formation of ribulose biphosphate taken place. Clear?
Yes.
Now what this RUBP will do? Again, it will capture a carbon dioxide and transfer it for the glucose formation. Got it?
Okay. So the same cycle repeats.
Right, right. So what is it? What is this? Like what is RUBP? RUBP is a transporter.
Okay.
It takes a carbon from carbon dioxide and deliver it to the glucose. Got it? Right?
Yes. Yes.
So every time it deliver 1 carbon atom, and for the formation of glucose it required 6 carbon atom. So how many time Calvin cycle have to operate, or C3 cycle have to operate for the formation of 1 glucose molecule?
Yeah.
How many time?
6 times.
Right, 6 time. Okay, now come over here. So I have one more question. So you are absolutely right. For the formation of 1 glucose, 6 time Calvin cycle have to operate. Calvin cycle have to operate. Now I have one more question for you, Affaf. How many ATP are required for the formation of 1 molecule of glucose? How many ATP and NADPH?
3.
How many ATP and NADP are consumed for the formation of 1 glucose molecule?
1 glucose molecule. 12 ATP?
No. One
No.
more ATP. One more ATP is there. In
There are
regeneration
2
process.
in like the first part, and
Yeah.
then there's one
One more.
in
Last
the
part.
third part.
So total three ATP in 1 cycle, and 6 ja 18.
So that's total 3. 18, yeah, 18.
Yeah. So
Not
18
12.
ATP. And how many NADPH? NADP? H?
2, 12.
Right. 12 NADPH. Right? So look.
Okay.
So total 12 NADPH. Here I have written over here. Right? 18 ATP are required in C3 cycle and 12 NADPH. And I have already told that the 1 NADPH is equal to 3 ATP. Right? So 12 into 3,
Yeah.
how many? 36.
36.
Right? So 18 ATP over here. Right? 36
Yeah.
ATP over here. How many total? 54.
54.
Right.
Okay.
So 8, 6, 4, and 1. Yeah, right. 54. So how, if someone asks you how many ATP are required for the operation of, or for the formation of 1 glucose molecule? Total 54.
54.
Did you got that balance sheet?
Yes.
Please write it down. You won't get it in any book. Please write down.
Okay, I'm writing it down.
Yeah.
Done, sir.
Done. Okay. So did you got all points?
Yeah.
Okay, fine. If you got C3 cycle, then we must go to the next topic, which has been covered in your textbook. So importance of the chloroplast, I have told you. Photosynthetic APR that I have told you. Chlorophyll. The photosystem that I have told you. Light dependent stage that we have explained. Jodi scheme that we have explained. Light independent stage of photosynthesis, we have discussed that. Using the product of photosynthesis. What is that? Uh, um, okay, so the factor. Okay, fine. Now we are going to study that the factor which uh, involve the or which affect the photosynthesis. That's what we are going to discuss. So that is the last thing which is mentioned in your notebook textbook. So the C4 cycle is not there actually in your textbook. So generally in CBSE there is a C4 cycle as well. One more cycle, but need not to mention. It's not there. The last thing that is this factor affecting the photosynthesis. So Affaf, there is a one law that is called limiting factor law. Just limiting factor a term have been given in your notebook. Limiting factor is not or law of limiting factor is not mentioned. So look. So can you tell me, Affaf, what are the limiting factors? It's a very common term, common English term. So what is limiting factor?
Anything that limits or affects photosynthesis
Right.
process.
In fact, I'm teaching you, you are studying. So between you and me, what is the limiting factor? You can say, sir, internet.
Yeah.
If internet is available for 30 minutes, 40 minutes, 1 hour or 2 hour, so that is the limitation. If internet is available for only 1 hour, then we can study only 1 hour, right? So between you and me, limiting factor is internet. While there are a number of the factors, laptop between you and me, right? The camera, studio, light, there are a number of the things. This is
Yeah.
stylus, everything, right? There are a number of the things. So the fan, AC, and all that. So these are the factors which are helping us to study. But every factor is available, but internet is available for 1 hour. So internet is limiting. Second thing,
Yeah.
internet is available for 24 hours, but between you and me, the power backup is available only for 1 hour. Now the power factor backup is the limiting factor, right? So the factor which is present in minimum amount, that is a limiting factor. And limiting factor is that which decide the rate of reaction. Got it?
Yeah.
Okay. So
Yeah.
first limiting factor is, if we call about the first factor is light. So in graph, here we can see, it's a bit messy. Let me write down and more clearly. So here we can see, right? Photosynthesis rate This is the rate of photosynthesis, and here this is the intensity of light. Intensity of light. So look, if intensity of light is increased, the rate of photosynthesis will increase, but up to a certain limit. We can see after that limit, if you will increase more intensity of the light, the rate of photosynthesis will not increase. That's how the light up, right? And if you will increase the light intensity very high, what will happen? Oxidation of the many of cell organelle takes place, and we call that process solarization, right? So did you got it?
Yeah. Yeah.
So light. Fine. So light is no more limiting factor because we know that light is unlimited. The second thing, if you want, please note it down. Solarization, please note it down. The term is solarization. First, please note down this graph, then I'll tell you what is a solarization. I explain.
It's done.
Done. So please note it down.
Yes.
Uh, when the light intensity increases beyond a certain limit, the oxidation of cell organelle, the oxidation of cell organelle takes place, and as a result, cell die.
As a result, what?
Cell die.
As a result, can you write it down?
Yeah, death. You can write as a result, death of cell take place.
Okay.
The process is called solarization. Next point.
Wait, sir. I have a question.
Yeah, sure.
You said the oxidation of cell what takes
Cell
place?
organelle. Let me write it down. Means the, like
Okay.
mitochondria, chloroplast. They all,
Organelle?
yeah.
Okay.
Organelle. Cell organelle. Means organ of cell.
Yeah.
You must have listened in cell biology topic.
Yeah.
So oxidation of cell organelle takes place. Means oxidation of chloroplast will take place, mitochondria will take place. Means the cell organelle will get destroyed. Means they will burn out, right? So
Yeah.
you can see that during the summertime, where the humidity is very like less, and due to high intensity of the light, many of the very delicate plant they die because of high light intensity. So that is the process where solarization takes place. Means plant dies due to high light intensity. Clear? So write it
Yes.
down. Yeah. one more point. Rate of photosynthesis increases. Rate of photosynthesis increases while increasing light intensity up to certain limit. After that, the rate do not increases. After that, rate do not increases while becomes stable. Next, carbon dioxide concentration. The next, let me check with your textbook. What is the next factor? Yeah, carbon dioxide. That's the carbon dioxide. Fine. So now it comes to the carbon dioxide. So the normal concentration of carbon dioxide in atmosphere in general, right? Normal concentration where we live. So that is a 0.03 to 0.05 part per million ppm, right? That is the normal concentration of the carbon dioxide. It is found around the earth, but in polluted cities or polluted area, it may be beyond that, or in like more pure area, it may be below that, right? But this is the general 0.03 to 0.05 ppm. So
So what is ppm?
part per million. Whenever we, count the, have you seen the pollution certificate of any vehicle, car or any other
No.
vehicle? No. Go through with it. You must have a pollution certificate with your car.
Okay.
So if you'll go through with it, there you will see that their ppm is written over there. Ppm is part per million, right? So whenever we count the concentration of any gas in atmosphere or anywhere, we use the ppm, right? Part per million.
Okay. So
Right?
0.03 is in what conditions?
Normal conditions, 0.03 to
Normal.
0.05, where we live, like in your room, in
Okay.
around your house, right? If it's not industrial area then.
Okay.
So okay. So what I was doing, yes. So and have you listened the term TQM?
What?
TQM.
PQ what? Oh, no.
This is the actually term for the purity of water.
Okay.
So if the TQM of water is more than 200, then this is polluted. It's not healthy for you to drink.
Okay.
But many of the cities, you will measure it, it reaches up to 1000. So this is the, we use the TQM, right?
Okay.
Okay. Now come to carbon dioxide concentration. If we increase carbon dioxide concentration, increase of the rate of the photosynthesis takes place. But for C3 plant, the increase takes place up to 450 microliter per liter. Please note it down. The normal concentration, the normal or average concentration of CO2 is 0.03 is 0.03 to 0.05 ppm. If we increase the concentration of carbon dioxide or if concentration of carbon dioxide increases, if concentration of carbon dioxide increases, the rate of photosynthesis increases. Full stop. The saturation point, of CO2 concentration for C3 plant is,
Sir, saturation point of carbon dioxide?
right, saturation point of carbon dioxide for C3 plant is 450 microliter per liter. 450 microliter per liter. Beyond that, rate of photosynthesis do not increases. Third point, temperature. What is the impact of temperature? Can you tell me?
If temperature is too high, the plant will not work very well because there's too much water
Temperature is
vapor.
high, then
Water vapor loss, I guess.
water evaporate, right? Very good. Yes, water scarcity takes place.
And as you said, cell organelles, they get oxidized. They die. They stop
Right,
working.
oxidation will take place. Right. That is also very true. So yes, rate of the photosynthesis depends on the temperature.
Rate of photosynthesis can take place. It can get higher if the temperature is higher, but after a certain limit, it doesn't.
If it's higher than,
If it's too high, then the cell will burn.
right, definitely solarization will take place, the leaf will burn, and the plant may dehydrate, right? It may dry up. And one more important thing, a very common question of the biology which I am going to ask you. So tell me, Affaf, suppose that now the winters are about to approach. So when the winters will come, sometime the winter reaches at the temperature reaches at that point in the minus, that people start dying due to freeze frosting. And even the same thing takes place in higher temperature. So either the temperature becomes too high or the temperatures become too low. Many of the people start dying, like including human and including some animals start dying. So why they die? Because of extreme
their
low.
enzymes get denatured.
Exactly, right. So that's all the temperature work, right? So temperature do not drop very much impact, but at high temperature or low temperature, or we can say at extreme temperature, the enzymes stop working, and that's how it affect the rate of the photosynthesis as well, right? You can write it down. Please write it down. The photosynthesis is maximum at optimum temperature. The photosynthesis is maximum at optimum temperature. Full stop. At extreme high or low temperature, at extreme high or low temperature, at extreme high or low temperature, enzymes stop working. At extreme high and low temperature, enzyme stops working, and enzyme stops working, and photosynthesis get affected. The same thing is water. Water is a limiting factor. Temperature can be limiting factor. Generally, CO2 and light are not limiting factor. Yes, but water is limiting factor. Optimum water is required for the photosynthesis. If the water scarcity will take place, the rate of the photosynthesis will decrease. So these were the factors which affect the rate of the photosynthesis. Now you can go through with your textbook, and in case if you have any query, you can ask me right now.
With my textbook.
And only this part of the textbook have been given to me.
Sir, there's this part chromatography in the
Okay,
same
okay.
topic.
That's very simple.
I don't think they've covered
Yeah,
that.
yeah. It's there. It's there. I've seen that. It was in chlorophyll, around chlorophyll. Yes, you know what is the paper? Do you know what is the paper chromatography?
I've seen that in my lab.
Yeah, you must have seen that in your lab. So what actually we do, we take a solution of the pigments, right? So chloroplast pigment, right? Or from the chloroplast we take out the chlorophyll, and we chromatograph paper. We took the put the chromatograph paper. Now the rate of diffusion is different of different color. Like carotene is highly diffusible, right? Then pheophytin, xanthophyll, chlorophyll A, chlorophyll B. So when you put the that chromatograph paper, due to rate of the diffusion, due to different rate of diffusion, they will reach at the different level. So on that chromatograph paper, you can, if you will put that chromatograph paper in that solution of the chlorophyll, you will find different these what to say pigment, right? So different pigment at the different part. So that's how, with the help of paper chromatography, we can prove that the chlorophyll is the combination of not only the chlorophyll B, it's a combination of carotene, pheophytin, xanthophyll, chlorophyll A, chlorophyll B, and some other pigment. Got it?
Yes, sir, there's in the same topic. Oh my God.
Yes.
There's this absorption and action spectra in the same
Yeah, that's why
part.
I told you that absorption spectrum. So rise between that lies between the 400 nanometer. You remember synthetically active radiation (PR), right?
Yeah.
So that is the absorption spectrum from 400 nanometer to 700 nanometer, and the graph reflect the same.
Okay.
You can see in this diagram, it's given over here in 400.
Light wavelength and
Right.
okay, so this has been talked about in our previous classes.
Yeah.
But then there's this RF value.
You are sorry. Once more.
There, in the textbook. Wait. So if you open page 9, you'll see it.
Okay, page number 9. Just page number 9. Yeah, page number 9. Yeah, I can see that. Yeah, tell me.
Yeah.
It's in front of me. So what are you asking?
The RF value.
The
RF
RF.
value.
Okay.
There's a
RF value. RF value. Fine. There is a nothing. Just the distance traveled by solute means the distance traveled by pigment. So RT values distance traveled by the solute and distance traveled by the solvent, right? So we measure both of the distance and we divide. That's how we get the RT value.
oh, okay. Okay. Other than that, I don't have any questions. In the PDF, I made sure to include the last pages of this topic, the exam practice ones,
Okay,
just so you know what kind of questions
fine.
are there in the exam.
Let me open.
They seem easier in this textbook, but they're much harder in exam.
Okay. Science communication. So those questions which has been given and question and in page number 19. No, this is the activity. No. Exam practice. This one 5A page number. It is given. This one exam practice.
Yeah,
The MCQ type of the question.
2021. Yeah, there are MCQs and the other ones, the graphs and the
Yes,
regular ones.
yes. I get. So MCQ, you can practice MCQ. The following substance involved in the photosynthesis, glycerol D3 phosphate. Yes, that we have discussed. And like how many molecule of ribulose biphosphate are broken down to produce 2 molecule of glucose? Can you tell me? Question number 3.
Question number 3. How many molecules?
How many molecule of RUBP are broken down to produce 2 molecule of glucose?
How many?
Yeah.
Oh my God. 2 molecules of glucose.
Easy. It's start like build concept, like start thinking like conceptual way.
Okay, I'll think.
Yeah. In one cycle of Kelvin cycle, one RUBP breaks down.
Yeah.
So for
I
one
guess
glucose
B.
12. Yeah, absolutely right. 12, that'll be the answer. So that's what I want to build the concept. That's how. That's why I was explaining. Most of the students, like 99.9% student, even in India, right? I'm talking about those students who are very brilliant. Like they get 96%, 97%, more than 97%. I have many of the students. They have secured 100% every time. Basically, 100% of them are girls. They are not boys in biology. So every time they belongs to very top school of Delhi NCR, and they get 100 out of. But out of them, leaving those students, most of the students think that biology is the thing to cram, and there they stuck. But what I say, the biology is the subject which you think logically, and as much as concept and logic you will develop, it will become easier that much for you. Okay.
Yeah. These questions are so much straightforward in this textbook, but sir, let me find a good past paper. I'll show you how confusing the exams are.
Right. No, sure. Yeah, tell me.
I'll share one.
Yeah, sure.
In the same email address, and I will send the next topic.
You can do one thing. You can share in this chat box as well. You can send with the help of chat box.
Sir, I tried last time. It didn't work.
Okay. Like. Go in chat box, right? And you can see there is a symbol which look like this one, a file like symbol, right? And from
no,
here,
I only see a smiley.
Okay, you can't see that. Fine. No issue. So share with me on that email ID. If I'll discuss with you in next class. No issue. So, but before going to the next topic, you have to share your some file or PDF with me, because this was I had this part of I had the PDF of this chapter only.
Yeah, I know. Actually, there are like 4 topics. The first 2 is equal to. There are like unit 4, 5, 6. I'm done with 1, 2, 3.
Okay. What is the
sixth
topic?
The 6th one is practical. Alternative to practical. It's
Fine.
done on paper.
Right.
So the first 2 topics are unit 4. The other 2 are 5. The 6th one includes all the practical questions.
Right. So share that PDF with me, then I'll discuss all those points, right?
Okay.
So should we wind up here today?
Yeah.
No, sure. So share those PDF, then we'll discuss in next class. Until the time, what I want to suggest you, go through with your those questions where you stuck, right?